separated and then recombining again. But we have two processes that we term them cis or trans. So in our studies, that is what we do. So when we talk of this cis and trans, before we do that, in molecular biology, we do genetic process. And we also use molecular markers also to track recombination routes in the genes. So when we come to the third one, we also look at, after using these two processes, we look at how the genes are expressed. So that is how we can use any of these processes to target cis and trans recombination in genes. So next slide.
Next slide.
Okay, so these are basic terminologies we must understand before we can appreciate the whole part. So when we talk of genes, we say genes are segments of DNA that contains instruction for building proteins. So before proteins can be built, they take information from our genes, and they determine the structure and the function of the cell. So next one, the chromosome. The chromosomes are structures within the cell nucleus that contain DNA, and they carry what? Genes. So they carry, these chromosomes carry the genes. So it is on the chromosome that we find the genes. So in the recombination process, they must there is a situation whereby some will be omitted or some will be added, and that is what we call mutation. So when we talk of mutation, it is whereby the DNA sequence of a gene is mismatched, and this DNA sequence, it comes in a specific order, and it must be in that order before it will be able to represent the character from the parent. So when we are looking at cis and trans recombination, we must understand this one, that DNA sequence must come in what? Specific order. Next one.
Put the example of plant.
Yeah, okay. So, we now want to look at the main subject for the day, at the cis and trans test for recombination in molecular biology. So, in molecular biology, we use the term cis and trans to describe the ability, orientation of genes or mutation on the chromosome. So, when we say orientation, we are looking at specific location where we are going to find that gene on the chromosome. So when we talk of cis, we are looking at genes that are found on the same chromosome, on the same chromosome. So that's why we call it cis. And it can recombine or swap genetic materials to enable attempts to get original state. So when they were combined, they separate again, then they recombine. So when those genes are on the same chromosome, they can use the neighbor cells to attempt to get back to the origin during meiosis. Next one is trans. Trans occurs when same or homologous genes exist on separate chromosomes. When we say homologous genes, we are talking of genes that are in two pairs, but they represent the same character. Let's say a gene that is coding for a height of a plant or human being. Height, we have two characters, either the plant or the human